Hello and welcome back to the Sanctuary. I'm your host, Professor C, and we're going to do a little bit of anatomy and physiology today. Again, specifically, I'm going to take us through what I'm calling an organelles lab, where I'm going to sum up all the organelles using a chart and showing some organelles on a picture and see if we can handle naming those structures. So let's do it. So here we go. Classic chart. I've given you the names of all the organelles / structures that are found on most of the standardized tests out there, right? Let's go through them very quickly. Function of a Golgi body. All right, remember there were 4. It was to, and I'll keep this short, receive packages from the ER, right? Modify those packages. I guess repackage would be what I could call that. And then deliver or secrete to wherever it needed to go into a cell or beyond. So 4 big things that the Golgi does: receive, modify, repackage, and deliver. Composition, it's membranous. I'm just going to put mem. It's a membranous organelle. All right, a nucleolus, not a nucleus. A nucleolus, this is where we synthesize ribosomes. Synthesize ribosomes. And remember, the nucleolus, because we're synthesizing ribosomes, is largely made of ribosomal RNA. Lysosomes. Function, we call them the janitors of the cell, right? So I will say they are digestive organelles. And we talked about them, how they could digest bone, how they could cause autolysis inside of a cell, so it will destroy itself, how it would get rid of foreign bacteria and viruses, broken organelles, and so on and so forth. So digest organisms and structures. Composition of a lysosome, it's a ball, right? It's a specialized vesicle, so I'll just draw a little ball. Mitochondria. Function, make ATP. Or you could say oxidative phosphorylation if you want to get super fancy. Or you could say does aerobic cellular respiration. That works as well. Now, compositionally, it kind of looks like a jelly bean with a wiggly inner membrane, right? So it is a membranous organelle as well. Cytoskeleton. Function, we could say give shape to a cell, give support to a cell, do a little bit of intracellular transport. Compositionally, it's usually protein, proteinaceous if you want to get fancy, long protein filaments, right? Microtubules, microfilaments made of protein. Centrioles functionally assist mitosis. By making spindle fibers, right, and pulling apart those chromosomes during anaphase of mitosis. So compositionally, a centriole is, remember they have the, I don't want to draw all of them, the 9x3, remember they have these 3 sets, or these triplets of long pipes, but they have 9 of them. 9x3 protein tubes, if I can fit it over here. Remember they have those long 3x3 by 3 by 3, and there's 9 sets of those 3 protein tubules. All right, flagella. Function, very simply, is movement. Unidirectional, one-way movement. Compositionally, protein. Long, whip-like protein tails. Rough ER, well, because it's rough, that means it's got ribosomes on it, so it has something to do with making secreted proteins and integral proteins, other things for the membrane, including phospholipids sometimes, and then packaging them up and sending them out to the Golgi. Compositionally, the rough ER kind of looks like ribbon candy with dots all over it, right? Those would be the ribosomes. So another membranous organelle derived from the outer membrane of the nuclear envelope. Ribosomes, of course, functionally make protein. and we talked about this in several different places, but just recently we talked about translation events and how they're super important in translating RNA into a protein structure. Compositionally, we know that they're made of rRNA. All right, peroxisomes. We call these kind of the hazmat crew, right? And I said if you could get it down to one word, detoxify is a really good word to use for peroxisome. Not just your normal janitors like a lysosome, but those that have some specific enzymes to neutralize bad stuff like peroxides. Compositionally, peroxisomes are like little balls, very much like a lysosome. They're membranous organelles. Vesicles, specialized vesicles. Cilia, function, move particles is an easy way to say it. They beat back and forth, and they help move particles that are adjacent to it, like in the respiratory system. Compositionally, they look like little hairs sticking up from the surface, right? But we know they're actually just little tiny protein threads, very much like little hairs. Smoothie art can do several things depending on what type of cell that it's in, including storing calcium, but most of the time it's about lipid synthesis. Even though, like I said, there are some other things you could put functionally, but lipid synthesis is almost a default answer on exams. So compositionally, smoothie art kind of looks like, the coral we drew. And even though it doesn't look as wiggly as the roughy are, it's still considered a membranous organelle. Grows off of the membrane. No ribosomes, though, stuck on this guy. It's smooth. All right, villi. Just remember they are the little fingers that stick up off the surface of a cell, and we may even see itty bitty teeny tiny fingers on top of the fingers. Those would be microvilli. But this has something to do with increasing surface area, usually for absorption or secretion. And there's a quick rundown of the most common organelles asked on standardized exams. Let's look at a picture and try to make our way through it. So let's see how many things we can name. If I let me change my color up a little bit. This guy here that I can scribble green, I like to do that. What's that guy? It's probably the Golgi body, right? A membranous organelle. We talked about their functions on the last page, but I would call that a Golgi body. All these little dots that are floating around, these little balls, I can't distinguish between which one's a ribosome, unless it's attached to the ER, of course, and which one's a peroxisome, which one's lysosome, and we're not going to deal with those little tiny ball-like structures. So let's see what else we can find in here. What about this line right here? It's pointing to this egg in the center of this, looks like spaghetti bowl, right? Well, if that bowl of spaghetti is the DNA and the nucleus, then that egg there is the nucleolus, right? And it's where the ribosomes are being made. And then what is this hole here in the spaghetti bowl? Well, that's a nuclear pore that lets things in and out. And of course, the spaghetti, all in here, the spaghetti is DNA, probably in the form of chromatin, but it could 10% of the time be in the form of chromosomes. What are these little guys? Looks like pieces of pasta, 90-degree angle, near the center of the cell. Those are the centrioles, right, that are going to assist with mitosis later on. If I drew a halo of threads around it, I could call it a centrosome. This fellow over here, perhaps P would be the line pointing to him. Well, it looks membranous, and it's got dots all over it, so I would call that the rough ER without even a second guess. Where's the smooth ER? Looks like it's right here, right? Kind of looks like coral, still bubbling off the membrane, but doesn't have the dots all over it, so it can't be rough ER. The only other thing that really pops out to me on this slide that's unequivocal is this. And that's that mitochondrion. It's got that smooth outer membrane. It's got those folded up cristae in the middle, and we should see multiples, and we certainly do see multiples of the mitochondria making ATP. So that's a good way of, if you can look at a picture like this and you can say that organelle is this and it does this function and it's made of this compositionally, and that organelle is this and it does this function and is made of this compositionally. If you practice this a while, you'll have them down no problem. All right, that's it for our little organelles review. See you for the next one. Bye-bye.